Okay, so this is the IACS Research Day, but we're actually here guests in the Laufer Center, right? So I don't know if all of you is registered on your brains, so this is actually the Laufer Center auditorium, and we're actually guests of Ken Dill here, who's going to be our inaugural speaker for Research Day. I'm sorry, the inaugural keynote speaker for Research Day. I'm going to call on Jeff Heinz now to formally introduce Ken.
Thank you. It's a real honor for me to introduce Professor Ken Dill to all of you today. As you may know, Professor Dill is a SUNY Distinguished Professor. He is the Lewis and Beatrice Laufer Chair of the Laufer Center, and he is also directs the Laufer Chair of Physical Biology, and he's Professor of Physics and Biology here at the university. Ken has studies protein folding, and he's won numerous awards for his research, including the Neurath Award from the Protein Society, the Max Dolbeck Prize from the American Physical Society, and also recently the Sacco International Prize in Biophysics. He's written textbooks. He works on the computational biology, and I'm also, one detail you probably don't know about Ken Dill is that he also worked with a mathematical linguist about 15 years ago. And he explored the possibility of applying generative grammar, the mathematical models of grammar, to the protein folding problem. So it's a real delight to hear him speak today about his research. Thank you.
I want to thank Robert, and I want to thank all of you for the invitation to come here today. And I want to tell you sort of what happens on this side of the wall between you guys and us guys. We're interested in computational biology. We are sort of a vision-driven center and a collaborative center. So I'm going to tell you about the work of sort of half of the work in the center is computational structural biology 2.0, we call it. It's about 4 of us here who work on this. And the case I'd like to make, I'm going to try to make, is that I think Stony Brook here in the Laufer Center is getting to a stage where we're sort of competitive with anybody in the business of deepening the math and physics underpinnings and pushing computational biology to the next level. And I'm going to try to actually make that pretty quantitative. So I'll show you my point here. We burn about, probably between Carlos and myself, I think we burn about 20 million hours on the supercomputer per year. But I want to show you that it's going towards something useful, not just burning energy. But one key point I want to leave you all with is our problems scale very badly, and we need a ton of help. And so despite what I'm going to tell you about some great progress I think we've made, we're not even close. We need help. So first of all, a protein, many of you know this, I'll go through it quickly, is a sequence of amino acids. Amino acids are single, small, simple molecules. They end up in 2 flavors, hydrophobic and polar. Basically, hydrophobic doesn't like water, polar do like water. The blue ones are polar, the red ones are hydrophobic. And so proteins tend to be this string of beads that glums up into a little oil droplet with the oil stuff in the middle. But different sequences fold up into different structures. And in your body, a human being, we have about 20,000 different types of proteins. So there's one sequence that folds to one structure, and that performs one kind of function. And then we have 20,000 of these different things. And so there's 20,000 different stories here. And we know probably in any detail, we probably know a grand total of less than 10 or less than 100, something like that. We have a long ways to go. And most of them are too big and complicated and fancy for us. So this is the point I want to make, is that every protein has a story. These are interesting ones. They're fun ones to watch. They're rotary motors like this, which are a big part of how you charge up your energy batteries. They're rotary motors like this that cause bacteria to swim. There are linear motors like this that move your muscles. There are pumps and channels like this that allow stuff through membranes. And then there's 20,000 minus 4 other ones that do other stuff. So what is it we want to compute? Our field, computational biology. Here's what we care about. First of all, we want to take that linear sequence of amino acids. Typically, they're 100 to 200 amino acids long. We want to take that sequence and we want to predict what is the folded structure. That's a hard problem, especially if you want to get the atomic details. We want to predict the binding and the motions and the conformations, because this is really the meat of what proteins do for a living. They perform all the functions in your body, and the functions are performed by doing binding and moving and so forth. And how do they assemble into bigger complexes? Proteins have this ability to hook together like Lego blocks into complexes that perform really big and fancy functions, collective machine actions. We also want to understand how they fail. This is a big part of drug discovery. Now, the point I want to make is that we need physics-based modeling to do this. And let me just draw a distinction for a second. There's a lot of deep learning that's going on in our world at the moment. I'll say a word about that a little bit later because there are some competitions where we know something about how well they're doing versus physics. But fundamentally, we're not going to really help any biologist until we solve physics problems. The data-driven things are just about looking at a protein and saying this is what it looks like. Trouble is, that's only the beginning metaphor for learning anything about how these things work. You got to have free energies, you got to have populations, and for that you got to do the hard work of physics, and that's really why we're trying to build that stuff here at Stony Brook. A lot of interesting stories about the history of computers, like the Blue Gene computer that IBM built, all headed, in my opinion, in the wrong direction because they thought it was the bigger hammer would solve the problem rather than better physics. And bottom line is, it's just the hard work of better physics. You got to have that. It's about mutations and environment. This is really all about evolution and superbugs and that sort of thing. How are we ever going to get there unless we can do the free energies and populations? You can't just look at structures for that. Folding processes, transferability across proteins. We have to avoid databases. We have a great database of soluble proteins, 150,000 proteins, atom by atom, every hydrogen atom. We know where it is. And you would think that surely should be enough for us to be able to go forward and figure out all the mechanisms. But the answer is there's a lot of proteins, including ones that are the most important sort of drug discovery targets right now, neurodegenerative diseases, Alzheimer's, Parkinson's. All of those diseases are going to be half of the GDP in 50 years from now. We have no idea how to deal with them. We also don't know how to deal with membrane proteins, which are sitting in membranes, and these are the things that have a big effect on that. So let me show you, this is familiar, I'm pretty sure, to most all of you here, is that we need an energy model to start from. This is a semi-classical model. This is the kind of thing that Carlos Simmerling works on and is really at the forefront of developing. These developed around, excuse me, around the 1960s, 1970s, thereabouts. And they've come handed down to us now in a generation and people like Carlos, for example, and David Case at Rutgers are the key keepers and developers of these models. We need a force field, so we need the parameters for these things. We also need solvent models. You heard Marivi's talk a little while ago on getting really deep into the quantum mechanics. The models, because we have so many degrees of freedom, hundreds of thousands of them, we have to put solvent into the problem as well. Because of the challenge of biology, the huge number, the size, and so on, we have to use fairly simple solvent models at the moment. We have to use either implicit solvent, where you just smear out everything, or we can use explicit solvent, but that's pretty expensive for us. So we go back and forth. So one thing is the energy model. Got to have that. The other key thing is we need search and sample methods. And this is probably pretty familiar too. Sorry, I don't know why it's cutting off up here. We need search and sample methods because this is how you get populations and free energies. And one point I want to make is there's only so far you can get in learning about proteins by looking at structures, because looking at structures can tell you something about energies, but it's what you don't see in structures that tells you about entropies, and most of biology is about entropies. And so the problem is, if you just go through and try to learn stuff from looking at structures, you're going to miss a lot of biology. It's driven by conformational entropy of the proteins, and it's driven by the solvent entropy. Anyway, we need things that do this well over very large landscapes in order to be able to ultimately get converged to the deepest minima here in free energies, and so those are the most populated states. Now there's good news and bad news here, and so I'm showing you two totally different things on this plot, but it really summarizes a lot of sort of where is the progress, where are we now, where have we come from. First of all, the blue line here is nothing but Moore's law. This is a classic result. All of you know this. On the right-hand axis is the logarithm of the transistor count in a certain amount of real estate on a chip. And this is showing you going all the way back 50 years to how that real estate density has improved. And this is, roughly speaking, it's a factor of 2 better density every 2 years. That's fantastic. It's usually taken as sort of the ultimate achievement in how fast you can make progress. But I want to show you this curve too. This also goes back about 50 years. This is the curve of force field simulations on proteins. It goes back to the original simulation of Karplus and McCammon in 1973. They did 3 picoseconds of simulation of a protein in vacuum. And this was a Nature paper. Of course, these days, that would be a joke. You couldn't possibly get this into anywhere interesting. But in those days, this is what they simulated, about 3 picoseconds. This axis is showing you the accessible time scale. It's also logarithmic. It's also over the same time. And you can see that it's very Moore's law-like, too. And in fact, if you really want to squint, you can see this slope on the red curve is steeper than the Moore's law curve. Now, that's not just hardware. That's also the force fields. The force fields have to keep up with the hardware. And so proteins used to fall apart in 3 picoseconds inside the computer because the force fields were crappy. These days, they don't fall apart in picoseconds. They don't fall apart until milliseconds. And so these are simulations up here. That red dot that you barely see, this is D.E. Shaw in New York City. What he did was, this is a guy that was a hedge fund gazillionaire, and he's building his own computers. He's built his own Bell Labs team, and he's been doing this for a decade now. And those guys are simulating pretty well because they've got some pretty amazing hardware. But still, having said all this, what I want to tell you is the problems we care about are right about there somewhere. So we're not there yet. But one good piece of good news is that for many years people said, well, you guys are doing molecular dynamics, you're still not designing drugs, you're still not folding proteins. Why is it taking you so long? And the answer is really in here too. And the answer is that nothing biologically interesting happens on a faster than a microsecond scale, except optical things in your eye, for example, but mostly folding and binding and all kinds of things that biology cares about. They happen on time scales that are up here and up here. So milliseconds to seconds. That's the biological time scale. And so the point about looking at these diagrams too is that we only have been there for about the last decade. So that means if I look at the progress that's going on in computational biology, I should start seeing in the literature over the past decade some really interesting problems getting solved. And the answer is we do. There's some really good news out there, and a lot of it actually is happening here. I'm going to tell you about that. So this is the group that I'm going to tell you about. It's Carlos Simmerling, Vigeli Koutsias, myself, and Dima Kosakov. And so again, we're aiming for trying to push computational biology, and we're trying to push it by solving the math and physics and computing problems. One of the things that's held this back is if you want to solve math and physics problems for biology, the trouble is you're dealing with siloed funding. NIH will pay you to solve a biology problem, NSF will pay you to solve a physics problem, but neither one of them will go into the middle and help you out. Fortunately, in the Laufer Center, we have some resources that have allowed us to go after some of these challenge problems. So Carlos's big expertise is in this software called Amber SB. The SB is for Stony Brook. It's a force field. It changes each year, improves all the time. All the rest of us in the group are using it because it's the best we can find. We try other ones too. And he's also a developer of solvation models that are good companions. I'm going to show you some results in a minute. Vigeli has worked on loop modeling and macrocycles. This is a big challenge too, and I'll come to that in a minute. And he's done an amazing job bringing mathematics to solving the problem rather than just wiggling stuff in a computer. We have an accelerator for MD that we can use for a lot of protein modeling, and Dima Kosakov has this method called ClusPro that is all about taking two proteins and trying to figure out how they bind to each other, because this is really sort of the fundamental unit of biology, is one protein talking to another to get stuff done. So a key thing I'm going to describe to you here are what are called community-wide blind tests. Probably the oldest, certainly the oldest one, and probably the one that's best known is this thing called CASP. And this has to do with protein structure prediction. It's now been in the business for about 25 years, and it has now provided a kind of a culture that other communities use too, not only us. There's, this one is about predicting protein structures. Capri now is about predicting how proteins bind to each other, and D3R is a solvation modeling competition. We have several of them in computational biology, but this idea of computer competitions that are community-wide is now much broader and out in the world. And honestly, it has really brought, it's put the feet to the fire of a lot of us in the computational business, because there was a lot of kind of garbage before, and the experimentalists didn't like it, and they decided to start holding our feet to the fire. So here's the way they do it. In CASP, for example, once every 2 years, in a window of about a month, what will happen is on a website appear amino acid sequences that nobody knows the structure of. So about 200 research groups will go back to their computers and use their models and their algorithms and see if they can fold up that protein. It's blind. They don't know what the answer is. Then a few months later, what happens is the CASP guys have done the experiment, and they tell you, person A was right, person B was wrong, and so forth. And this is at atomic detail. It's a very refined business now, so you can say a lot about the details of the structure, details of the algorithm, how transferable are they. So these are blind tests, and they're community-wide competitions, and they're very useful because we learn a lot by comparing our algorithms to each other. And I'm going to show you some examples from this. First example I'm going to show you is Carlos's force field. He did an outstanding job in this competition called D3R. I think this was fall of 2018, and I think the results were in December. What they did in this competition is a series of something like 39 different ligands. This is an example of one of them, binding to a protein. It was not known how these things bind and their affinities. This is the 20-odd groups or more that competed in this competition. The y-axis here is showing you the correlation of the computation with the experiment done blind afterwards. And Carlos is the 2 in yellow here. Turns out he did better than everyone else. This is his amber force field 14. He's got now amber 18. Is it 8? You're up to 18. Every couple of years, he adds more refinements and developments. One thing you can't see, unfortunately, I guess it got chopped off at the bottom here. I noted a thing that really is, to me, super impressive. The amber 14 force field of Carlos gets 700 citations per year. Most of us would be thrilled to get 700 citations across everything we do in a lifetime. This is a very popular force field. Everybody likes it, and it's because it's really good, and you can see that here. I'm going to show you DEMA now. DEMA is, Kosikov is, it says automated protein docking. His algorithm is called ClusPro, and what it does is it computes the binding of 2 proteins to each other. This is showing you kind of what you know. What you know is the structure of this protein, you know the structure of the blue protein, and what you're trying to figure out is how do the 2 of them come together, and what's the orientation of them, that's called the pose, and then what's the free energy of binding of them. Right now the free energy of binding is out of the range of any of it. None of us can do any of that stuff. But at the moment what you can do is test whether your algorithm gets a decent pose or not. What DEMA brings to this is fantastic because he does this on a lattice. He makes these shapes on lattices that represent the 2 proteins. He does systematic global sampling on the lattice. These are with physical potentials. And he accelerates this by a sort of a rotational and translational fast Fourier transform method. So his method is very fast relative to the others, but it's also now, he's been developing this for a while, it's also very good. And to make that case, let me show you, this is the blackboard that was shown, I'm sorry, this is not showing up. This was from Capri 2019, this was just last week. This was the blackboard that they showed in Hingston, UK, which is where they did the Capri event, where they did the evaluation of the Capri event. And what you can see is ClusPro came out number 1. This is DEMA's method up here. What I have down here, which you can't see, sorry about that, is that he has something like, ClusPro server has something like 20,000 users. This too, in the protein-protein interaction world, trying to figure out how 2 proteins interact. This too is kind of the dominant algorithm that's now out there. It's very successful at getting these things right. It's very heavily used by users. All right, next up, I want to tell you about macrocycles and loops. This is the work of Vigeli Koutsias. Macrocycles, this is an important area. They are big, complicated molecules. And when I say big and complicated, what I mean is not relative to proteins. They're much smaller than proteins. What I mean is relative to normal drugs. Normal drugs are relatively simple, small drugs that have molecular weights less than 500 for reasons that have to do with how do you get drugs into the body and so on. But right now, there's sort of a third world of drug discovery that's just about coming online. The first world of drug discovery was pharmaceuticals, normal drugs, aspirin, stuff you take it by pills that you eat by mouth, for example. The second world of drug discovery was biologicals. These are big antibodies, and these are the drugs that now, some cancers you can cure with them, but they cost you $400,000 a year to do it. They're big, expensive, complicated drugs. And the biologicals, or the biotech drugs, you have to inject them because the trouble is they're big and complicated, and if you just ate them, you would break them down with your proteases. Your proteins would chew them up. So those are the two worlds that are out there now in drug discovery. The small molecule pharmaceuticals that you take as pills, the large molecule biologicals that all the biotech companies produce. There's a middle world that's largely been sort of out of range of any of us so far, and that's the macrocycles. And they have a special niche. In certain ways, they're a lot cheaper. In some ways, they are better from the point of view of things like side effects and so on. But nobody's tackled them. They're very hard computationally because you can see there's a lot of degrees of freedom in this, and they're constrained and complicated degrees of freedom. So they've been too complex and floppy for molecular dynamics, but this is showing you a list of things that are already known to be either drugs that are made by companies, by pharmaceutical companies, or these are toxins that biology itself makes because it knows that it can make really effective molecules out of these, like cyclosporin, for example, to prevent transplant rejection, and antibiotics of various types, and biological toxins and things of that type. So the issue is, can we address macrocycles? And what Vigeli does is he does this better and faster than other computational methods. This says better, faster loops and cycles. He uses a method he calls Bricard, named after a geometer, I guess, from the 1890s or something like that. It's called inverse kinematics, and what he does is he solves these. It's like, imagine sort of a bunch of rods all connected together. If I push on this one, how does that one move? You can do a lot of that analytically, and some of it you have to do by sampling. But by virtue of being able to do a lot of it analytically through his geometric methods, what it does is it speeds these processes up enormously relative to what anybody's been doing in the past. So I'm just going to, you can't see these very well, but this is a speed performance comparison. This is log of time, and all of these black dots here are showing you various tests he's made, which are about 2 orders of magnitude faster than finding macrocycles structures by using just normal molecular dynamics that currently is the state of the art. He gets structures that are much better. This is a 0.3 angstrom structure rather than versus 4.9 angstroms, which is the current best. And then there are some that are so big and complicated they've been unsolvable, and he's able to get them. One thing I think is really beautiful about these kinds of this approach he's got is the bigger and more complicated the molecule is, usually, as you would imagine, the slower it would be to compute. It's just big and complicated, and you just got to move stuff around. In his case, because he's got the math of constraints helping him out here, the bigger and more complicated it is, the faster it often can be. And that's very powerful. We care a lot about this. For one thing, loops. He applies this to loops in proteins, and loops are important because that's often where the business end of biology happens. Is there's some motional part in the protein, and some drug will bind to, in that region. But in addition, it's also about macrocycles. So this is really sort of about next generation pharmaceuticals. And he has gotten together with Dima. Whoops, sorry, that disappeared. What it says is Dima plus Vigeli win in the macrocycles competition in D3R in 18. And again, that's a competition. The results just appeared last week. That was also, I guess, in Hingston, and across 25 groups. And this is showing you, in this case, the RMSD error of these blind predictions, and this is showing you all the groups that competed, and this is showing you Vigeli and Dima down here, and this is an example of a ligand in a protein binding site where they got the right answer before anybody knew what the right answer was. I'm going to take a few minutes now to tell you about some things from our group. And perhaps I probably have a few too many of those slides and a few too few of everybody else's just because I know our work. But this is called MELD Can't read that. MELD accelerates molecular dynamics. And so the basic idea is molecular dynamics right now, the physics methods in general, are just much too slow for most interesting biology. Best I can do with a protein with straight molecular dynamics is I can wiggle around the very smallest proteins, and I can learn a little bit about some motions of them. But ultimately, we need much bigger horsepower. So our aim is to put in the physics, that is molecular dynamics, put in external information of some kind, and have that gracefully integrated in a way that we can get structures and actions. Now the problem is, okay, I got it on this slide now. The problem is that this area of putting sort of data in along with physics and molecular dynamics is sometimes called integrative modeling. It brings together whatever form of data you have with physics. The problem is that when you use integrative modeling, it's very graceless. It doesn't handle the data well. So you put in the external information, but it has to be correct and accurate and unambiguous and sufficient, and even experimental data is rarely good enough. But we even want more vague and combinatoric data. And so the problem is we have messy data, and it's hard to get that data to help inform molecular dynamics enough. So the method that was developed here by Justin McCallum and Alberto Perez, who are both now off in faculty jobs. This says Bayesian funnel approach of MELD, and what it does is it takes a needle in a haystack problem. I'm looking for a needle, and I've got a huge haystack, haystack being conformational space, the needle being the lowest free energy conformation in conformational space. And what I do is I'm going to break this haystack into sub-haystacks. And I'm going to have many, many possible sub-haystacks. And so each sub-haystack itself is very searchable. And I can do that with a Bayesian approach in which I have basically one prior, which is the Boltzmann factor for the whole force field for all of its conformations, and then a whole bunch of likelihood functions, each of which is relevant to a different sub-haystack. Now, how do we choose the haystacks? How do I parse the haystacks? And the answer is I actually use the force field to do that. So rather than having the data tell me where the MD should look, the MD tells me which data I should pick that's consistent with it. That's the basic idea here. Let's see, metaphor. A metaphor would be something like this. Imagine I just tell you there's some treasure box key somewhere in Stony Brook, and it's in a gas station. Well, then the only thing that you know is that I better look around gas stations, and that's the sub-haystacking. I'm going to go to near gas stations, and every time I'm near a gas station, now I'm going to look around, and then I'm going to see if I can find that key. And so the thing is, there's the information that you know, in this case, is coming from the force field telling you, okay, look here, okay, look here, okay, look here. So I'm going to show you a few examples of what we do with MELD. It's a pretty good accelerator, sometimes up to 5 orders of magnitude, and it depends on what information you have and what you're looking for. One thing that we've done with it already in the past, in the last couple of years, is use MELD to help experimentalists take their data and determine a molecular structure from it. There have been other tools that do this. MELD turns out to be pretty good. I'm not going to show you slides about that because it's already 3 years old. But what I am going to show you is the sort of tip of the iceberg of structure determination. These days, the really exciting area of protein structure determination is called cryo-EM, won the Nobel Prize last year. But the trouble is the cryo-EM, this is electron micrographs basically getting protein structures. That's normally way too small. But by taking all these various slices, you're able to get enough information that often you can put it together and get protein structures, especially for big, complicated proteins. Now, the problem with it is the data is very limited and sparse. It doesn't actually specify the whole structure for you. It doesn't specify the whole structure for you, and so you need a computational model that can take very limited data and fill in the blanks, and that's what MELD does. This up here says converting cryo-EM to structures. And we're working now, okay, sorry, wow, you can't see the bottom here. The bottom says, points out that this is a collaboration between our group here, Alberto Perez, the developer of MELD, who's now at the University of Florida, Klaus Schulten's group at the University of Illinois, he's not there anymore, but his group is still there, and one of his former students who's now at Arizona State University. It's a 4-way collaboration. Those guys have been able to, the Illinois guys and the Arizona State guys know how to take data from cryo-EM and put it into molecular dynamics, but it's not been enough to give them structures. And now we add MELD to all this, and it does turn out to be enough to give us structure. This is an example of a structure. Their methods are over here. This is their called MDFF. It's their molecular dynamics package. You take you have a way of converting cryo-EM densities into the MD code, and then we use MELD in an iterative sort of cycle here to be able to refine structures. And we can get to reasonably sizable structures. We can get to membrane protein structures. We haven't published this yet. This is sort of new. We're also trying to work on using small angle X-ray scattering, where again, the data is way too limited under normal circumstances, and MD won't get you there by itself. But MELD maybe can do it. We'll see. Let me tell you about a different, rather than structure determination, let me tell you about protein structure prediction. And what I mean by that is, what we're now trying to do is, I don't have any data at all, let's say. Somebody just says, hey, here's an amino acid sequence, good luck. And so what we do is we take these heuristics. They're heuristics like this. We call them vague directives. I want to tell my MD code, make me a hydrophobic core, or make me some nice secondary structure, or be pretty compact. Well, that's way too vague. And this is showing you the combinatorics of the make me a hydrophobic core problem. All the red lines, here's the amino acid sequence here that I start with. I don't know what the structure is. And every red line is showing you a possible pairing of two hydrophobic residues that could be coming together into the native structure. Now the problem is, if you go look in the database and look at lots of protein structures that are known, what you find is, of all the possible hydrophobic pairings, only 8% usually get made. But the trouble is you don't know which 8%, so I don't know what to tell my MD code. This is a combinatoric mess that I can't get it to weed through, but this is the kind of thing that MELD is really good at. What I tell MELD, instead of saying, oh, make this one, or make this one, or something, is it's a little more like statistical mechanics, this Bayesian thing. What I tell MELD is, here is the success ratio, here's the success rate. And in that case, I put in 8%. I just say, okay, MELD, 8% is what you're going to get, and go for it. And it just tries a whole bunch of things. And then the way this MELD method works is it's doing a conformational search in MD. And then as it starts to get into structures that it likes in terms of its free energy, it says, okay, what is the data that I'm consistent with here? And then it picks the best springs, if you will, that are the data constraints, picks the best springs and says, okay, those best springs are now going to try to make a structure. And then I do that for all these other sub-haystacks. So it turns out we can fold proteins this way, small proteins. We call it MELD XMD because it's a MELD accelerator of MD, and it folds small proteins from sequences using these vague physical directives. So here's what we did. We took these 20 small proteins. These had been run before from D.E. Shaw in New York City, and at the time he was sort of the only one that really could access molecules of this size. He didn't get a lot of them right, but he had the most horsepower and computer power. But now with MELD, we can leap over a lot of the hardware issues. So we fold about 15 of them to good accuracy, and we miss about 5 of them. What MELD told MD was make a hydrophobic core. We know that's about 8% right. Make secondary structures. We know databases give that about 70% right, and make it compact. And so it's pretty good at doing this for small proteins. We did this also for a set of proteins we call unthreadables. These are interesting because here's one of the problems is that people now use databases. And so if you take an amino acid sequence, they say, this looks a lot like lysozyme, and I already know the structure of lysozyme in the database. I'm going to use lysozyme to tell me where this helix is, and so on. So a lot of this is all the methods that are done in CASP right now are all database methods, with the exception of this MELD MD. And we can now bring physics up to the level and the speed that's needed to actually fold proteins. So these are ones where the databases, there are database sequences in there, but it turns out they're misinformed. They're so misinformative that they drive you in the wrong direction. And so it turns out when you put physics in instead of databases, you can get the right answers in many of these cases. A performance test. These are brute force numbers from D.E. Shaw for how long the MD takes. This is the logarithm of the folding times. These are the folding times we get in MELD for these different cases. And you can see it ranges from 2 or 3 to 5 orders of magnitude at speed. And the scaling is pretty good. This is the figure that kind of shows the problem we have with Moore's law. What we do is even recognizing that the force fields in the hardware are improving at a Moore's law rate. The problem is now let me convert that to the protein problem I have, which is to say protein size, when am I going to be folding proteins of a certain size? Right now, Shaw or we can fold proteins in the size range of around 100 amino acids. But MD, normal MD, scales so badly that if I want to get to sort of reasonably interesting proteins, pharmaceutically interesting, I need to be up at least 140 amino acids, and I won't be there for another sort of 40 years. But with the MELD scaling, it turns out we'll get there a lot quicker. We should be there in about another 5 years or so. One of the powers of this MELD XMD method is that we get all the good stuff from MD, meaning we get populations, we get real free energies. And that's because at the end of the day, the MELD springs are pulling on stuff, but at the end of the day, when they're done pulling, you're at the bottom of an energy well, and the springs have sort of have totally disappeared. So we can expect that we might be able to get the method to tell us when the method thinks it's got it right. All of the prediction methods in the past, nobody ever knew when they had it right. You would get to mail in your 5 best predictions, best meaning whatever you think is best, mail them in, and then if you ask the predictor, which of your 5 do you think is the best one, can you rank order them? The answer is no. A lot of papers were written on the fact that essentially it's random. Once a predictor says, here's my best predictions, and it's random sort of which order they come in. But we can do this with populations because we get free energies now. So it turns out indeed when we get converged populations, which happens still mostly only for small proteins, can't get to big ones still. But for small proteins, this is showing you populations that we're getting. If the populations are low down here, then the bottom line is we don't think we're doing any good. But we know that in advance before we mail this into the predictor, to the CASP. But here, when our populations are converged on a single one, then that's when we know we think it's right. This is a case where we are now able to bring physics, 5 minutes, okay, thanks. Now able to bring physics to CASP, and we get right answers, and we get best in CASP in some cases. I won't go through the details of that. This one is where we did the same. This was the CASP that just ran last summer. These are 3 cases where the MELD MD method gives us best in CASP again. Let me skip this one. This was an NMR refinement, and MELD XMD is best in CASP for refining NMR structures, taking NMR data and giving you structures. I'm going to go through this rather quickly because the stories are pretty simple and similar. This is, we can do binding of fairly complex ligands and compute rough binding affinities, 2K plus or minus 2 kcals. Not terribly great, but it's better than nothing, which is what we've had in the past. We can get reaction coordinates. I'm going to end with this one. I think this is the last example. We can start applying this to some disease problems. This one says cystic fibrosis up here, which is a protein problem. It's different than a normal drug discovery problem. In normal drug discovery, pharmaceutical companies know what to do. They want to try to make a small molecule that binds to a site on a protein. This is not that kind of problem. And all the neurodegenerative diseases are not that kind of problem either. So all the pharmaceutical companies a few years ago got out of the neurodegenerative disease. They're not even chasing those problems, despite the fact that it's a huge, whopping market, something like 4% of GDP in a couple of years. But they're out of that market because they don't know how to deal with them. And this is an example of that kind of problem. This is one, too, which is a protein conformation problem or stability or motion problem. This is the protein that's the problem in cystic fibrosis. It's a channel. Chloride goes out. The chloride, when the chloride is properly going out, what happens is the mucus out here does not get clogged up. On the other hand, when you don't have a proper channel, the mucus gets clogged up and you have a lung problem. And so this is a problem of inserting that protein getting it out through the Golgi apparatus and getting it into membranes. What we know from our simulations, and this is really only because MELD accelerates MD enough, is that it looks like it's a motional problem. Not emotional, but a emotional problem. This one is wild-type protein. This one is the disease mutant. It's a mutation at residue 508. And this, it turns out, are called restoring mutations. It turns out if you get the disease mutation and you get an additional second mutation, it turns out you're okay. And so the question is, why is that? And all that's been known is sort of just the fact that these restore. This makes you sick, and this one is healthy. But we can get into the microscopics of it, and we can see the variance. That is the motions here. Let me just end right here and say, so summary is that what we're doing is aiming for better math, physics, and computational methods. For biology, still have a long ways to go, but we have a really good team here. So Carlos and his gang do force fields and solvation, modeling, searching and sampling is happening in a lot of places, Vigeli and us, and also DEMA. We are now, a bunch of companies have been interested, so we're working with these companies on various kinds of problems like this. And it allows us into a new realm of drug discovery, the neurodegenerative diseases like the Alzheimer's, Parkinson's, Huntington's, cystic fibrosis, that sort of thing. Still, there's a long ways to go on all of this stuff, but it allows us into that realm. My last slide is just a point I want to make. This is a paper that Al and I wrote, and up here are all the problems that the deep dive data methods are giving solutions to. Here was Deep Blue, the solution to chess. Here's AlphaGo a few years ago. Here are the corresponding sizes of proteins down here by numbers of degrees of freedom and the combinatorial complexity. And so we were folding these teeny little proteins about the size about when Deep Blue was solving its problem, and they had about the same number of degrees of freedom. So we're in about the same place. The AlphaGo problem